So this particular audience doesn't need a huge amount of background on the fact that we have mammal declines in Australia. There was particularly some of the earlier declines were largely in arid areas. And then there's been these ongoing declines more recently in Northern Australia. So that's the kind of backdrop of this project. That they're happening. That I think that the reasons for the declines are, so we've been talking about feral predators as the main driver, but that doesn't necessarily exclude the possibility that there might be other drivers, right? Go to the next slide. So as I mentioned, the historical mammal declines happened largely across most of the arid zone. We had these huge contractions of species that were distributed right across the country, contracting right back to tiny areas, slightly more mesic areas, and all going extinct completely, right? And then more recently, from about I'm always lost for time, but certainly since about 2005, it's been clear that there's been declines happening in Northern Australia, and there's been a few little hints that there might have been some spatial patterns for those around declines travelling in a westward direction. I only know this because I've been working on cane toads at the time, and I think if the mammal declines had have happened, 12 months later in some places, we might have been thinking it was cane toads that had done it. So there was this clear pattern of declines, or that looked to be hints of a spreading kind of pattern to it, and that got me thinking about disease quite a long time ago. Next, if we go to the next slide. And so I've been thinking about diseases generally for many years, and of course have been involved in work on chytrid fungus, which, as we all know, that has been implicated in the declines or extinction of hundreds of species of frogs across the world. But there's also other examples of diseases that have swept through populations, essentially wild populations, and caused mass mortality and major impacts. And in particular, what needs to happen for those diseases to really drive populations extinct, which seems to be what's happening, certainly in the case of chytrid, is that the disease needs to be to have a reservoir host, right? So normal disease dynamics, like for example, is what is playing out with humans at the moment. If the disease has used up all of its host population, then it declines or it goes extinct, right? And that used up can be either an immune response or death of the host. But if you're a generalist, and particularly if you've got reservoir hosts, then you can potentially persist in reservoir hosts whilst having this kind of ongoing, continuing impact on another host. And so that's the kind of scenario where we might have a disease that moves through a place, sits in reservoir hosts, often not having a major impact on them, but can be crossing species to other hosts where it can be having a major impact. And those hosts that are being very badly impacted, they obviously have the risk of going extinct. So I've been thinking about this for quite a long time, and that made me think. So there's a million possibilities, right? There's a million possible diseases out there that could cause disease, and many of them we simply don't know about. Wildlife diseases are not well known, but one that struck me as being a distinct possibility was toxoplasmosis. If we go to the next slide, please, Holly. So, Toxoplasma gondii is the name of the pathogen. It has a really complex life history, but it is a generalist, so it definitely ticks the generalist box. It affects a huge array of organisms, right, from earthworms through to humans, and its primary host is cats. And we know that cats are implicated in declines of mammals anyway. So I think, here's potentially another means by which cats might be having an impact. Now, it has this really complex life history. I'm just going to step you through it quickly so you're all on the same page. Cats are the primary host. That means that's where sexual reproduction happens. So a cat, an uninfected cat, or a cat that's never been infected, a naive cat, ingests the oocysts, so the eggs from the environment. They get into the cat's gut and they proliferate wildly. And they form a sexual form in the cats. and that goes to form eggs, right? And so for a few weeks after a cat is first infected, it's shedding eggs into the environment. After that, its immune system kicks in, and the parasite goes, oh crikey, let's get out of here, and it goes and insists itself in all of the body tissues, right? So it just forms these quiet cysts. In other species, so everything, as I said, from earthworms to humans, the cysts are either picked, so the eggs are either picked up, or you can also catch it from these other cysts, environmental cysts. You can pick them up, again, they go into your gut, and again, they proliferate wildly, and then you have this acute infection where you can often feel very sick, and this pathogen is going crazy in your gut. Once your immune system recognizes it and starts to fight against it, it does the same thing. Goes from proliferating wildly in the gut to getting into the bloodstream and just insisting everywhere. So that's what it does. It's got this really complicated life history. But importantly, for the reasons I was interested in it, or we were interested in it, this is Nalika also, is this idea that actually it could be sitting in cats as a kind of uninfected, the cats aren't affected by it very badly, and it could be seeding out into the environment where it's affecting lots of native species. And there's lots of, there are many anecdotal stories essentially of acute toxoplasmosis killing native Australian animals. So everything from wallabies through dunarts and rats. And these are observations that have been made in captivity. Also releases of bandicoots, for example, on French Island. They had a whole bunch of bandicoots become infected with toxo, just picked it up in the environment and died while they were radio tracking them through acute toxo. So you can have this issue of during that process when it's proliferating wildly in the gut, you can have this acute toxoplasmosis phase, and that many native Australian animals, or native Australian mammals, have been recorded to have died in that process, right? So we've got a pathogen that we know kills things. We know it's got a reservoir host. We know it's an incredible generalist, and we don't know a huge amount about what strains we have in Australia. Nor do we know very much about its distribution in Australia up until recently. So that was the backdrop for the project. Let's go to the next slide. I think I might have already talked about the next slide. Oh, so the other thing I wanted to mention was that when it gets into this, so it's an interesting pathogen in that when it gets into the cyst stage, when it insists in the host body, it's been well documented to have these kind of sublethal fitness effects. And that's a kind of complicated way of saying it. It can change individuals' behaviour. So there's lots of studies showing that people that have toxo tend to take greater risks. There's also a few classic studies showing that rats that have been infected, that are in this kind of chronic phase with toxo, are actually attracted to the smell of cat urine instead of repelled by it. So there's clear evidence that this pathogen manipulates its hosts in interesting ways. It can also skew sex ratios of offspring, right? So one of the arms of this project that we certainly started looking at and attempted to look at was this idea that actually, even if it doesn't cause acute effects, there may be sublethal effects that change sex ratios or change behaviour in ways that might go on to affect population fitness, right? So we might not be seeing acute effects from toxo, but it could still be having an impact on populations. So questions about where it was in Australia and what it was doing to our mammals were what drove this project. Next slide, which I've already talked about. Yeah, so let's talk about sublethal effects. So this was a really tricky project to pull off. So we started this project again. Nalika Hughes and a PhD student started this. Turned out to be logistically very difficult to get the toxo cultured up, but we had great help from Chris Tonkin around that. The other thing that turned out to be very difficult with the ethics around this. So essentially what we wanted to do was to give these model native animals a sublethal dose or a sublethal infection of toxo, get them into that chronic phase, and then and have a control group where we didn't have that happen to them, and we measured their behavioural differences. So next, the next part of the project was more about trying to find out where in Australia they're from, right? And so there was, it turned out there was, our target here was to look at the primary host cats, right? So one of the issues with prevalence in wild populations for any pathogen is that you might find zero prevalence, right? So you might go out and sample your bandicoot population and find there's no individuals for toxo. If you were to conclude that no one gets infected by toxo from those data, that would be a very poor conclusion to reach, because in fact it might be that actually 90% of the population was infected and they all died, and as a consequence you don't see them, right? So prevalence is a very crude measure of prevalence in hosts that are affected and potentially have experiencing pathogenic impact from the parasite. Prevalence is a bit dangerous, right? So instead we decided to focus on the primary host, which is cats. And cats don't suffer very badly from having toxo, but it's their primary host. It's the host that all toxoplasmosis cells are trying to get into. So we figured it was a good organism to focus our efforts on. We did a big literature review, and we found lots of work, actually from some of which was funded by previous incarnations of NESP and various other people around the place that had found that had done little bits of work about prevalence of toxo in cats. So we put all those together, and we also contacted people all around Australia because we knew that there was a massacre of cats going on across the country, and we wanted to try and get as many samples as we could from people. And we got quite a lot of buy-in from a lot of groups. And again, Nalika Hughes did all of the comms work around that, about reaching out to people and trying to convince them to post us pieces of cats. In the end, we accumulated an additional 314 samples from 277 individuals, and we pushed them through a PCR detection process and scored whether that individual had or did not have cysts, toxo cysts in the tissue, basically. What that process revealed, so not only did it massively increase the number of sites that we had represented, and you'll notice that we didn't do a great job of getting into Queensland. We tried pretty hard, but didn't have much success getting samples from Queensland. So there's a big blank spot there. But what our data was able to do also was to compare across tissue types, to compare whether the size of the tissue sample mattered, and whether the sex or the age of the cat mattered. And to a first approximation, the answer to that is that it didn't matter too much which tissue type we sampled. It didn't matter too much how much tissue we sampled, certainly within the range that we sampled. And it didn't matter too much what the sex or the age of the cat was. So it turns out that cats, if they're going to be infected, often get infected quite young with toxo. So we, so that's the sample size we had to work with, but we didn't find any really obvious big effects, the kind of things you might expect to affect detection to affect it very much. That gave us a bit of confidence to put our data together with the data that we got from the literature to bring together a kind of large data set across 50 sites around Australia. And the overall prevalence across the country from that data set was 38% of cats had toxo. So pretty good, actually, pretty high prevalence. So it's a reasonable, it's where you want to be working from a statistical perspective, right? Is close to the middle of that range. So that's what the raw data look like essentially. Holly, if we can move to the next slide. Oh God, I promised I would never put a table like this. I would strongly advise my students never to put a table like this into a presentation, but this is here mostly for my benefit, to jog my memory. Essentially what we did was we used these data to put together a model describing the prevalence in cats. And there's been some good literature done on the survival of the eggs in the environment. The survival of the eggs is strongly influenced by temperature, moisture. So cool, moist sites or cool, moist conditions, the eggs can last for months to years. Hot, dry conditions, the eggs don't last very long in the environment at all. So we use these variables of temperature and precipitation as proxies for temperature in the environment and water in the environment, because we knew that these would affect the survival of eggs, right? And so we knew that these should drive variation in transmission. The other two variables that we thought were important was urbanisation. So all environmental variables become decoupled from the greater world in an urban environment. And we also had this great layer that Sarah Legg and co had put together about the density of feral cats across the country. So we put those in, those four things in as explanatory variables to see if we could explain prevalence in cats. The model ended up getting very complicated because there was lots and lots and lots of interactions. Essentially, what it boils down to is that there was one model describing what happens in urban areas, and there was another model that describes what happens outside of urban areas. And outside of urban areas, things happen as you would expect. As temperature increases, prevalence goes down. As precipitation goes up, the prevalence goes up. So these would have been predictions you would have made from first principles about egg survival, and they're playing out across the continent. Certainly that was the inference we made. Interestingly, the effect of feral cat density was not very strong. In fact, it was about as close to zero as you could hope it to be, except in urban areas. So the model kind of indicated that feral cats are an issue in urban areas. So certainly the density of urban areas with a higher density of feral cats have a higher prevalence of toxo. The other thing that was interesting about the model was that it clearly said that urban areas have lots of toxo, right? So feral cats that we've sampled from urban areas have high levels of toxo. And as I said, it's largely decoupled. Yeah, thanks, Holly. That's largely decoupled from the broader environmental picture. So this is the broader environmental picture when we put this out on a map. So we basically take that model and we turn it into a map. And what you can see is that, you know, our best predictions for prevalence is that it's high in all the kind of cool mesic areas of the country, and really, really low across central and northern Australia. What this map doesn't show you is the urban areas and the fact that there'll be hotspots around lots of urban areas throughout the country, right? So most of the infected animals that we got from Central Australia, for example, or from Northern Australia, were around communities and urban areas. When you go out in the bush and look for toxo in those areas, you don't find it. So the picture that emerges, at least in my mind, is that we have, you know, urban areas are these kind of these places with high prevalence of toxo across the landscape. But by and large, the parts of the country that have experienced the big mammal declines, by and large, they're not good parts of the country for toxo. So next slide, I think that's on the next slide. Yeah, so to my mind, I think that has enabled us actually to rule out the possibility that toxo was a driver of historical and contemporary declines in northern and central Australia, which is not the result we expected, but it's kind of good. It's quite conclusive, actually. We could say toxo, it's probably not a concern. It's probably not caused the decline in central Australia or been involved with it in any way, probably, and similarly in northern Australia. So we can rule that one out. That's, you know, the frustrating bit about that is it still doesn't kill the disease hypothesis, but we can certainly rule out what seemed to me to be quite an obvious candidate. The other conclusions we'd make are that there's clearly high prevalence in cool mesic areas and around urban areas. The issue, you know, and we know that acute toxoplasmosis kills animals, and we know that it happens in the field as well.